You can grow and develop solely because of the replication of one molecule, deoxyribonucleic acid, or DNA. How does this work? The structure of DNA. We have gone into the structure of DNA in more detail in the 2022 special video. You can go check it out afterwards. In summary, there are four basic building blocks of DNA: adenine, thymine, guanine, and cytosine. Adenine and thymine, and cytosine and guanine, pair together through specific hydrogen bonds. The two strands of DNA are antiparallel, meaning that they go in opposite directions. The two ends of each strand are called the 3' and the 5' ends, depending on the number of carbon atoms. In a sense, this asymmetry creates a preferred direction for DNA, but in the opposite direction relative to each side. Organisms need to grow and develop, and this means that cells need to copy themselves. We have already gone into the detailed story behind how cells divide in a previous video as well. In short, cells need to make a copy of everything inside of them before dividing, which includes their genetic material. So the DNA inside each cell, all 2 meters of it, somehow needs to be copied, ideally without any mistakes. How does it do that? How to replicate DNA. As you can imagine, copying DNA is much easier when the two strands are separated than when they are tightly coiled up together. So to begin the process, cells somehow need to find a way to separate the two strands and carefully multiply each of them. Luckily, cells have a secret weapon: enzymes. Enzymes are specialized proteins that help speed reactions along without being changed themselves. They work by binding to a specific chemical, called the substrate, at their active site, which fits together like a key into a lock. An enzyme called DNA helicase begins the process by breaking the hydrogen bonds between complementary bases. Essentially, helicase is a zipper that opens up to two strands of DNA. As the DNA unzips, it forms a giant Y shape, which is called the replication fork. Topoisomerase prevents the coiled sections from becoming too entangled, while single-stranded binding proteins, or SSB proteins, prevent unzipped sections from coming back together. And from the replication fork are two strands of DNA, the leading and the lagging strands. Let's examine how each of them are replicated. An enzyme called primase produces a piece of RNA, called the primer, which attaches to the 3' end of the leading strand and acts like a landing pad. DNA polymerase, an enzyme that can pair up nucleotides, binds to the primer and begins reading the existing nucleotides on the leading strand. Incredibly, DNA polymerase can match up to 749 nucleotides a second. Because DNA polymerase can only start at the 3' end, it matches the complementary strand in the 5' to 3' direction. That means that it moves in the 3' to 5' direction of the original template strand towards the replication fork. Since the DNA polymerase is moving in the same direction as the helicase, it synthesizes the new strand continuously. Meanwhile, things are a bit different on the lagging strand. Because the lagging strand runs in the opposite direction as the leading strand, the complementary strand that DNA polymerase has to form runs away from the replication fork. Similar to what happened earlier, the primer binds to the 3' end, where the DNA polymerase begins matching up nucleotides. However, at the same time, the helicase continues unzipping and widening the replication fork, meaning that there are now parts of the lagging strand that have not been copied. To solve this, another primer attaches itself to the new 3' end of the lagging strand, beginning the replication process again. The synthesized strand fragments are called Okazaki fragments. This cycle repeats over and over again until the original DNA strands are completely unwound. After the DNA polymerases have finished pairing up the bases of the lagging strand with a new complementary strand, the primers are removed. To fill in the gaps left behind by the primers, another enzyme called DNA ligase glues the gaps together. Conclusion: although the gargantuan universe, with its billions and billions of galaxies, may seem to be the only source of complexity, if you look close enough, there are multiverses right inside of you.